Dear viewers, this video is part 1 of the two-part series on blood groups transfusion in the Medicine for Anyone in the YouTube channel. Blood types transfusion. Both blood types and blood groups mean the same thing. The surface of the human erythrocyte is covered with hundreds of distinct antigens. Some of these antigens are proprietary to RBCs, but others are found on organs, tissues, and fluids throughout the body. Transfusion of RBCs requires assessment of antibodies to RBC antigens that are circulating in the recipient's blood. Antigenicity causes immune reactions of blood. When blood transfusions from one person to another were first attempted, immediate or delayed agglutination, that is clumping of RBCs, and hemolysis, that is bursting of cell membrane of RBCs, of the red blood cells, that is RBCs, often occurred, resulting in typical transfusion reactions that frequently led to death. Soon it was discovered that the blood types of different people have different antigenic and immune properties, so that antibodies in the plasma of one blood type will react with antigens on the surfaces of the RBCs of another blood type. If proper precautions are taken, one can determine ahead of time whether the antibodies and antigens present in the donor and recipient blood will cause a transfusion reaction. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Major red blood cell antigens. Of the many hundreds of antigens described, none are more clinically important than the ABO blood group antigens. The A and B antigens are defined by immunodominant sugars N-acetyl 3-galactosamine for the A antigen and D-galactose for the B antigen. The A and B antigens are expressed on a precursor antigen called H. In blood group O individuals, H is the only expressed antigen. The expression of the A and B antigens on RBCs is weak at birth, but full expression occurs within the first few years of life. Naturally occurring antibodies to the A and B antigens are generally present 3 to 6 months after birth and reach adult levels by early childhood. In addition to RBCs, the A and B antigens are also expressed on vascular endothelium, lung, heart, liver, kidney, and bone tissues. Because genes controlling ABO and human leukocyte antigen HLA are inherited independently, major blood group antigens need to be taken into account in stem cell as well as solid organ transplantation. Note: location of ABO blood group. The expression of A or B antigens and thereby an individual's blood group is the result of allelic combinations of genetic variants in the ABO gene on chromosome 9, that is in the location 9q, that is long arm 34.2 region. Major histocompatibility complex MHC system. This system is known as the human leukocyte antigen HLA in humans, located on the short arm of chromosome 6, that is P6p21.3. Multiplicity of antigens in the blood cells. At least 30 commonly occurring antigens and hundreds of other rare antigens, each of which can at times cause antigen-antibody reactions, have been found on the surfaces of the cell membranes of human blood cells. Most of the antigens are weak and therefore are of importance principally for studying the inheritance of genes to establish parentage. Two particular types of antigens are much more likely than the others to cause blood transfusion reactions. They are the OAB, that is ABO system of antigens, and the RH system. OAB, that is ABO blood types, A and B antigens agglutinogens. Two antigens, type A and type B, occur on the surfaces of the RBCs in a large proportion of people. It is these antigens, also called agglutinogens, because they often cause RBC agglutination, that cause most blood transfusion reactions. Because of how these agglutinogens are inherited, people may have neither of them on their cells, they may have one, or they may have both simultaneously. Major OAB, that is ABO blood types. In transfusing blood from one person to another, the blood of donors and recipients is normally classified into four major OAB blood types as shown below, depending on the presence or absence of the two agglutinogens, the A and B agglutinogens. When neither A nor B agglutinogen is present, the blood is type O. When only type A agglutinogen is present, the blood is type A. When only type B agglutinogen is present, the blood is type B. When both A and B agglutinogens are present, the blood is type AB. Genotype OO, blood type O, agglutinogens nil, agglutinin anti-A and anti-B. Note, the type O blood, although containing no agglutinogens, does contain both anti-A and anti-B agglutinins. Genotype OA or AA, blood type A, agglutinogen A, agglutinin anti-B. Note, type A blood contains type A agglutinogens and anti-B agglutinins. In other words, when type B agglutinogen is not present in the RBCs, antibodies known as anti-B agglutinins develop in the plasma. Type A blood contains type A agglutinogen and anti-B agglutinins. Genotype OB or BB, blood type B, agglutinogen B, agglutinin anti-A. Note, type B blood contains type B agglutinogens and anti-A agglutinins. Genotype AB, blood type AB, agglutinogens A and B, agglutinin nil. Note, type AB blood contains both A and B agglutinogens but no agglutinins. Genetic determination of the agglutinogens. The ABO blood group genetic locus has three alleles, which means three different forms of the same gene. These three alleles, IA, IB, and IO, determine the three blood types. These alleles are called A, B, and O, but geneticists often represent alleles of a gene by variations of the same symbol. In this case, the common symbol is the letter I, which stands for immunoglobulin. Note, allele is one of two or more versions of DNA sequence, that is a single base or a segment of a base, at a given genomic location. An individual inherits two alleles, one from each parent, for any given genomic location where such variation exists. If the two alleles are the same, the individual is homozygous for that allele. The type O allele is functionless or almost functionless, so it causes no significant type O agglutinogen on the cells. Conversely, the type A and type B alleles do cause strong agglutinogens on the cells. Because each person has only two sets of chromosomes, only one of these alleles is present on each of the two chromosomes in any individual. However, the presence of three different alleles means that there are six possible combinations of alleles as shown in the above paragraphs: OO, OA, OB, AA, BB, and AB. These combinations of alleles are known as the genotypes, and each person is one of six genotypes. A person with genotype OO produces no agglutinogens, and therefore the blood type is O. A person with genotype OA or AA produces type A agglutinogens and therefore has blood type A. Genotypes OB and BB give type B blood. Genotype AB gives type AB blood. Relative frequencies of different blood types. The prevalence of the different blood types among humans are O 47%, A 41, B 9, AB 3. It is obvious from these percentages that the O and A genes occur frequently, whereas the B gene occurs infrequently, and AB is the least frequent. Agglutinins. When type A agglutinogen is not present in a person's RBCs, antibodies known as anti-A agglutinins develop in the plasma. Also, when type B agglutinogen is not present in the RBCs, antibodies known as anti-B agglutinins develop in the plasma. Note that type O blood, although containing no agglutinogens, does contain both anti-A and anti-B agglutinins. Type A blood contains type A agglutinogens and anti-B agglutinins, and type B blood contains type B agglutinogens and anti-A agglutinins. Finally, type AB blood contains both A and B agglutinogens but no agglutinins. Titer, that is concentration of agglutinins at different ages. Immediately after birth, the quantity of agglutinins in the plasma is almost zero. At 2 to 8 months after birth, an infant begins to produce agglutinins, anti-A agglutinins when type A agglutinogens are not present in the cells, and anti-B agglutinins when type B agglutinogens are not in the cells. The maximum titer of agglutinins is usually reached at 8 to 10 years of age, and this titer gradually declines throughout the remaining years of life. Origin of agglutinins in plasma. The agglutinins are gamma globulins, as are almost all antibodies, and they are produced by the same bone marrow and lymph glands cells that produce antibodies to any other antigens. Most of them are IgM and IgG immunoglobulin molecules. But why are these agglutinins produced in people who do not have the respective agglutinogens in their RBCs? A possible answer to this question is that small amounts of type A and type B antigens enter the body in food, bacteria, and other ways, and these substances initiate the development of the anti-A and anti-B agglutinins. For example, infusion of group A antigen into a recipient having a non-A blood type causes a typical immune response with formation of more anti-A agglutinins than ever. Also, the neonate has few, if any, agglutinins, showing that agglutinin formation occurs almost entirely after birth. Agglutination process in transfusion reactions. When bloods are mismatched so that anti-A or anti-B plasma agglutinins are mixed with RBCs that contain A or B agglutinogens, respectively, the RBC agglutinate as a result of the agglutinins attaching themselves to the RBCs. Because the agglutinins have 2 binding sites, IgG type, or 10 binding sites, that is IgM type, see discussion on lymphocytes immunity in Medicine for Anyone in YouTube channel. A single agglutinin can attach to 2 or more RBCs at the same time, thereby causing the cells to be bound together by the agglutinin. This binding causes the cells to clump, which is in the process of agglutination. Then these clumps plug small blood vessels throughout the circulatory system. During the ensuing hours to days, physical distortion of the cells or attack by phagocytic white blood cells destroys the membranes of the agglutinated cells, releasing hemoglobin into the plasma called hemolysis of the RBCs. Acute hemolysis occurs in some transfusion reactions. Sometimes, when recipient and donor bloods are mismatched, immediate hemolysis of RBCs occurs in the circulating blood. In this case, the antibodies cause lysis of the RBCs by activating the complement system and forming a membrane attack complex, also called cytolytic complex, that inserts itself into the lipid bilayer of the cell membranes. See complement actions in lymphocytes, the immunity in Medicine for Anyone in YouTube channel. This insertion creates membrane pores that are permeable to ions and causes osmotic lysis of the cells. Immediate intravascular hemolysis is far less common than agglutination followed by delayed hemolysis, because not only does there have to be a high titer of antibodies for lysis to occur, but also a different type of antibody seems to be required, mainly the IgM antibodies. These antibodies are called hemolysins. Dear viewers, this is the end of part 1 of the two-part series on blood groups transfusion in the Medicine for Anyone in the YouTube channel. Thank you for watching.